Okay, I'm going to introduce our next two presenters who are from SUNY Oneonta. Claire McMahon is a master's student at SUNY Oneonta studying lake management. She has a bachelor's degree in environmental sustainability from Oneonta and 4 years of experience in habitat restoration, aquatic and terrestrial invasive species management, and environmental education. Claire is spending the summer as a watercraft inspection steward team lead for the Catskill Region Invasive Species Partnership. And Melanie Rooney graduated from SUNY ESF with a degree in aquatic and fisheries science and marine science minor. She worked for the Chautauqua Lake Association, who's here today, obviously, and SUNY Oneonta in 2022 by participating in the herbivore and macrophyte surveys. In February of 2023, she formed her own business, Rooney's Aquatic Biologist, where she continues her research by monitoring the 5 biological control agents to limit the growth for Eurasian water milfoil. She does that work in 3 lakes, Chautauqua, Bear Lake, and Lake Bonaparte. So they're going to talk to us about SUNY Oneonta's Chautauqua Lake Macrophyte Survey.
Hello, I'm going to talk to you about the application of the point intercept rake toss relative abundance method on Chautauqua Lake. As you just heard, I'm Claire McMahon. I'm a lake management graduate student at SUNY Oneonta. I study aquatic plants and more recently freshwater pearly mussels. I'm also a boat steward team leader for the Catskill Regional Invasive Species Partnership. So I'm going to jump right in. Photosynthesis is what allows for aquatic macrophytes to grow in lakes. And for this to happen, you need water, nutrients, and light. And for nutrients, this comes from a variety of sources like fertilizer, bird droppings, soil, especially for waterfowl. Canada geese are really widespread throughout the United States. And this is why it's really helpful to have a buffer strip planted around lakes to mitigate some of this nutrient input. So some photosynthetic organisms that you can find in lakes. We have aquatic plants. These are really important to aquatic environments. There's a wide variety of aquatic plants found within Chautauqua Lake. We also have freshwater seaweeds or macroalgae. This includes like Nightella or Chara, you may have heard of. And then we have microscopic algae. There's a wide variety of these also, and these can be beneficial, but in the form of HABs or harmful algal blooms, which we've heard a bit about today, they can be quite detrimental. And this is an algal bloom on Chautauqua Lake, and this one is also. So aquatic plants are quite easy to kill. The hard part is finding something to fill the spot that they leave when they die. So when plants die, invasive plants can easily take over because they tend to thrive in these disturbed environments. So Eurasian water milfoil, I'm sure you've all heard of this. This is an aquatic invasive plant. It's present in Chautauqua Lake. And there's been some concern over it. It's found widespread throughout many lakes and rivers throughout New York State. In most of these water bodies, it has remained unnoticed. It is able to find a balance within the native community, and it's not a problem. But when there is some kind of upset of the balance within the water body, this is when it becomes out of balance and the milfoil can take over. So some of these disturbances can include introduced sediment into the water body from mud flows, road salts, herbicides, and also carp. These are all things that will weaken the native plant community. And so milfoil is a little stronger than the native plants, so anything that would weaken them, it allows milfoil to take over. So long-term monitoring is important because it allows us to see trends over time, and we can use these trends to make predictions for the future. And the more data you have, the more accurate the trends are. And you can use these to make better management decisions and practices. So monitoring plants specifically is important because plant communities change a lot, and this is season to season and also year to year. So it's a good idea to be keeping an eye on the plant community and the assemblage so you can know if things are in balance or if things are getting out of whack. If you know there is an invasive species present in a lake, it's a good idea to keep checking on it to know, is it staying in balance, is it spreading, is it decreasing, etc. But plants are really important in aquatic environments. They limit shoreline erosion, they improve water quality, they sequester nutrients, and they can hold nutrient-rich sediment from tributaries, and all of these can mitigate against cyanobacteria and nuisance algae. So I'm going to talk a little bit about PERTRAM, or Point Intercept Rake Toss Relative Abundance Method. And this is what we've been using to study the plants on Chautauqua Lake to assess the assemblages. So PERTRAM is probably the best method that you can use. It's relatively easy to teach to people. It's also easily observable by lake stakeholders, and it can be done in a reasonable amount of time. So this is better than some other methods that have been used. The only real limitation for PERTRAM is with the invasive species Hydrilla. It's a plant, and it's quite delicate, so often will shatter when it makes contact with a rake, so it could be undetected or underdetected. So on Chautauqua, when we do a PERTRAM survey, we'll do 2 rake tosses at each location, and then each rake, whatever we get up, we will assign it a category based on the abundance that we see. And often we'll get native mussels up on the rake also, and so we make a note of that also. And these are pictures of mussels that were found during last year's survey. So when we pull up the rake, these are the different sizes, categories that we will assign to the abundances. There's 0, obviously that's 0 plants. And then there's trace, which is fingerful. Sparse is about a handful. And then medium or moderate is a rakeful. And dense is pretty much too big to bring into the boat. Yeah. So PERTRAM is it's a lot more useful than just a presence-absence survey because you can assign a number to it and you can have more information about how things are changing year to year by the specific species, not just as a whole. And this also helps implement best management practices. So this is more of a breakdown of the different categories that we assigned to the rake amounts. And also we have the pictures on the sides. When we make a map of what we find, we'll have these little colored squares along at each point. So it's an easy visual to look at a map and see what plant is where and what abundance. And we also have a rough estimate for the dry weight, so you can have an idea of the biomass of plants that you're pulling up. So when you bring a rake in, you'll make an assessment, a visual assessment of it, and assign it a category, one of the letters that we just talked about, and then you will sort it into the individual plant species that you've pulled up and assign it a percentage. So you have an idea of how much of each species there is. So here's an example of what a map would look like after a survey. Each square is a point with the associated color that we went over. This is for curly leaf pondweed. Typically you'll find this in the spring and early summer, and by fall it has died off. So you can see it's comparing it from spring to fall of the same year, and then you can also look at the same species from a year to another year. So this one is spring, fall, in 2019 and 2020. And you can also use this for native and non-native species. There's a couple different ways that you can view the data. This is really useful because you can just look at the map and see areas of concern, if there's like a hot spot where invasives are being introduced, or you can watch things spread along the shoreline. So it's a good visual. And this is just a larger image of the one that was just on there. This is the 2022 survey data. This is just the North Basin. We have the native and then the non-native over here, plants and algae. And then this is the same 2022 native versus non-native, and this is within the Southern Basin. And this map is specifically Eurasian water milfoil, north on the left and then south on the right. And this is another way to visualize the data. This was made in R. Shows over spans a couple years, seeing what this trend is for Eurasian water milfoil. We have the North Basin and then the South Basin. So the North, relatively stable, it's decreasing a bit, and then the South is increasing. And this is just the presence of Eurasian water milfoil, not abundance, as in the amount of sites that it has been found. So takeaways from that is that the probability of finding Eurasian water milfoil is decreasing in the North Basin and increasing in the South. And this is for a Chautauqua specific model. So these are this is the data from the 2023 survey. The one on the very bottom, if you can't see, that's Eurasian water milfoil, and this is the amount of sites that has been found, not the abundance. This is just native biomass. We have the South on this one, and then this is the North. The biomass for both, for 2023. And then this one is by year. For the whole lake, not by basin, we have 2023 and then 2022. So some key takeaways from everything is that Eurasian water milfoil is the most prevalent species by occurrence, as in it was found at the most number of sites. Coontail has had a marked decline. This is due to herbicide use, and coontail is a native species. From 2022 to 2023, the total macrophyte biomass has decreased by site. Starry stonewort, this is an invasive macroalgae. It was found at 7 sites in 2023. This was last year's survey. And then the main takeaway is that native biomass has decreased while non-native biomass has increased. And that's all I have for now, so I'm going to switch it over to the other presentation.
I'm sure I can figure it out.
There you go.
Oh, perfect. Ooh, it's not at the beginning. Let's go at the beginning.
You got the presentation.
There's the little button. Sorry about that. There we go. Hi, everyone. I am Melanie, and I'm the new owner of Rooney's Aquatic Biologist. I'm here to talk about the importance of herbivory on Chautauqua Lake. Let's see. There we go. Herbivore survey started in 2002 with Racine Johnson Aquatic Ecologist, and we've been able to continue it till present day with myself being able to continue his work. So what we'll do is every year we'll go to these 15 locations. We have 4 in the south and 11 in the north. Our newest location is in BB. We added that one in 2017. So what we'll do is we'll go to each of these locations and collect 25 samples of Eurasian water milfoil at each station. And then we'll collect these and put it in a bag, and we'll take them back to the labs and observe it under a dissecting microscope. We'll see what insects are growing there, what damage they've done, put it I'll record it on a sheet, and then put that information in an Excel or Access spreadsheet so we can analyze it for further analysis. So over the years, we have found that Chautauqua Lake has 5 herbivores that help control Eurasian water milfoil naturally. These include the weevil, a moth, 2 types of caddisflies. We have a larger caddisfly and a smaller caddisfly, and a damaging midge. The weevil is probably the most important insects out of these 5 that I'll be talking about today. They're the highest in numbers, and they are the most effective with controlling Eurasian water milfoil. So what the adults will do, here's an example of what the adult looks like. I'll say they start their season on shore, underneath the soil and leaf litter. What they'll do is come springtime, once the weather starts warming up, they'll move from this shoreline into the water. They have to use native plants sort of as a bridge or a connection layer to get to the milfoil. Once they get to this milfoil, here's a great picture of it. The adult will lay their eggs. Now their eggs are really hard to identify. They are squishy, they're small, they look like sand. What do I know? What am I looking at? So what we can do is they're a little yellowish in color, and they have this black eye spot. You can see it on right there. It's hard to tell on this picture, but if you're not looking at an eye spot, you're not looking at an egg. So it kind of goes hand in hand. Once these eggs hatch, they develop into these little larvae. You can identify them best by their dark head capsule. Their bodies tend to have different colors, so that's a little bit trickier to tell. So if you find that dark head capsule, you got a weevil larvae. These weevil larvae will mine in the stem of the Eurasian water milfoil. Here's an example of 2 of them I pulled out of this stem. So they'll actually go inside of the stem, eat it completely, come back out when they're done. Then once they're done growing, they will then form a pupal chamber right here. And this is their development stage from a larvae to an adult. And this is almost the start of a fully mature adult. You can start to see their little eyes and their nose as they're becoming back to the adult. And they're able to grow and reproduce all season long till fall time. Then they'll go back on shoreline to overwinter underneath that soil and leaf litter. Next we have the moth. A little bit of a different life cycle than the weevil. The larvae, here's an example of what a larvae looks like. They actually overwinter underneath the ice and underneath the freezing water inside of the stems of the milfoil. So they persist in the water all year round, all winter long. Once it's warm enough, they will come out and develop into the adult. The adult will lay anywhere from 100 to 300 eggs, and you can identify these little guys by they have this little crisscross or figure 8 pattern on their face so that you can't confuse them with something else. These eggs then turn into larvae. That larvae also has that crisscrossing pattern on their face, so you can identify them very easily. They have 5 instars as larvae, so they'll be growing and getting bigger as they go from one instar to the next. They then form a cocoon where they are developing back into the adult. And if we're lucky throughout the year, we'll be able to see 2 or 3 generations throughout the season before they have to go back into the stem and overwinter for next spring. Next we have the larger of the 2 caddis. This is the walker or ghost caddis. They also have a slightly different life cycle compared to the weevil and moth. The adults require Eurasian water milfoil or curly leaf pondweed. The little flowers stick up in the water in the springtime, so if you're looking out and say, oh, what's these little flowers sticking up? It's most likely Eurasian water milfoil or curly leaf pondweed. But these caddisflies absolutely need these flowering plants to be able to lay their eggs. Once these eggs are laid, the larvae will once again feed heavily all year round, turn back into adults to start the process all over again. I'm not going to go through the life cycle again for the small caddis. They're generally the same. We have a little bit less information about the small caddis because they're a little bit of a, I'm not going to say newer to Chautauqua Lake, but we only have started recently finding populations of them. And they can be found in really high amounts. I have found over 100 individuals on one stem, and we only collect about this much milfoil at one time. So you can imagine over 100 little of these guys, as you can see right here, how packed they can get together while they're eating and consuming the milfoil. So they'll actually start from the bottom of the plant and work their way all the way up, consuming the entire leaflets of the plants. As you can see here, the little leaflets are scraps. Yep, these little parts right there are supposed to be the leaves that the caddis have already consumed and eaten. On this picture, it's right here and here and here. And then on this picture, it's all of these. Here, 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 yeah. Sorry, they're both green, so it's a little hard to tell leaves versus caddisflies. And lastly, we have a damaging midge, also present in the lake. These guys are probably the hardest out of the 5 to identify, because once we bring them back to the lake, they lose a lot of their color. So if you're lucky enough, like I was last year, I found one finally after all these years. Bob Johnson took this one about 20 years ago. You can see the little crisscrossing figure 8 pattern on both of these pictures. That's the best way to identify these. There's a lot of other non-damaging midges in the lake, and it's really easily, they're confusing to identify a lot of the times. What these little guys do is they'll actually create little retreats with the leaflets of the milfoil. They'll kind of crisscross them and make them a little home for themselves, and then they eventually eat the top part of the milfoil as well. Unfortunately, they are the least effective due to low population numbers. And I'm not going to go through the life cycle. It is the most similar to the moth. The results of last year are different than other years than we've seen before. For the past 6 years or so, we are generally seeing a decrease in biomass, not abundance, biomass of milfoil. So with this decrease in biomass, I have a small visual for everybody. So normally I take 375 samples. This year I could only take 213. These were all the ones I could not take. It was about a 40% decrease from all of those 15 sites. And we were not able to collect at locations F or M, and then we were only able to collect a small amount at locations A, D, G, I, and BB. And then overall, since we are seeing this decrease in biomass of milfoil, we are also seeing a decrease in the amount of weevils and caddisflies present in the lake. Excuse me 1 second. That's better. So here's just a general overview of the decline of just weevils. I don't have these fancy maps for caddisflies yet. We're still kind of developing that information. And I just want to turn your attention to our location A, which is in Burtis Bay, all the way at the southern point. It's actually right by the house with the smiley face on it. And in the beginning of the surveys in 2002, when Bob Johnson was doing it, we were able to find about 2 of these weevils per collection, per stem. Currently, if we look the last 3 or 4 years, I'm finding 1 weevil to every 2 or 3 stems. So it's just getting a lot harder to find these guys overall. Why is this happening? First and foremost, we are finding less milfoil. The density of milfoil is going down. So they're having less food to consume, and they're also having to interact with each other a lot more. This picture, this picture showed up a little bit blurrier on here. This is a picture of an essential larvae having to interact and compete with resources with an adult weevil at the bottom here. Another reason why they could be declining is there is mechanical harvesting that happens every year, but if you look at the picture in the back there, they show how mechanical harvesting works. They really only cut off the top parts of the plants, which leaves the bottom parts of the plants. So that's a great thing for these insects. They can retreat down to the bottom of the plants and survive and last long term. Another recent development that's been happening for the last 6 years or so is that herbicides have been being used from about the Bemis Bay area all the way down to the south. And we are seeing impacts on locations A, B, G, I, and BB. And then with these recent herbicide usage, we are also seeing slightly more dense algal blooms, specifically more in the southern areas than in the northern areas. And this could cause the lake to shift from a clear water state full of macrophytes to a turbid state full of algae. So in conclusion, these 5 little insects are the best way to control Eurasian water milfoil all naturally. It's a great alternative to using these other toxins like herbicides. And the weevils specifically help promote the growth of native species because they will not grow or reproduce on any other species besides Eurasian water milfoil. And Chautauqua Lake is a little bit of a catch-22 right now. So we're seeing these declines of weevils and caddisflies, but we're still seeing Eurasian water milfoil in the lake. So what do? We're turning to herbicides to help treat these areas of the lake, but unfortunately they're targeting other species as well, and along with killing these insects along with it. So if we keep using these herbicides, what's going to happen to these insects in the long run? But unfortunately, nobody's commercially selling these herbivores to help manage the lake in the long term. So what do? We keep on trying until we figure something out. And with that, I would like to thank the Chautauqua Lake Association that has been able to help me continue this valuable work. And unfortunately, Bob Johnson had to retire a couple years ago, and he has been an invaluable resource to be able to continue this work. And I've been able to have all his data, and he still consults with me. I saw him on Monday, actually, so he's still able to contribute in that way. And with that, are there any questions?
There's a question here. I'm just trying to make sure I can read it. It's getting at so what's the ideal range of plant biomass, specifically in the littoral zone? They're giving an example. For example, what percentage is too low and what percentage is too high? Fish kills from plant respiration or normal plant decay. That's the general idea. And if I've missed something key in that question, please let me know.
For a Percham survey, we typically say medium to sparse is a healthy range for a lake to have for plants. I don't know if you got anything.
Yeah, that's pretty much what I was going to say, yeah.
There is another part of the question.
What was the other
I think this is the same. It's the same handwriting, so I think it's the same question. What's the impact of mechanical harvesting on native plants such as coontail and on the spread of invasive species? And also the impact on herbivores.
I'll talk about this a little bit more. We actually did a small survey on the harvest. Oh, sorry about that. We actually did a small survey with Racing Johnson, aquatic ecologist. I'm sorry, I forget which year we did it. And we actually went out with the harvesters and did a small survey to see how much gunk or leftovers or whatever you want to call it goes, like when they're collecting the plants from the harvesters, how much gets left over and off the boat. We found a 0.6% of loss, 0.6, less than 1%. So when they're collecting these plants, they are collecting almost all of them, which is amazing, I thought. So the harvesters are not spreading any invasive species because they're able to collect almost all of it at one time. And I don't have any correlation between native plants and harvesting. If we're seeing a decrease with that, I don't believe so, just because of the sheer nature of harvesters only trim the grass or cut the lawn, as everybody keeps saying. They don't take out the whole plant altogether. So even though you're cutting the grass of coontail, or actually you can't cut coontail. Coontail is free floating, so that's not an issue either. Say like Elodea goes really tall and to the surface. Well, if you only cut down a meter, you still have 2 more meters of Elodea left. So you're not getting rid of the plant altogether, and you're still able to have a healthy ecosystem, and the insects can still survive as well because those insects at the bottom 2 meters didn't get taken out. But we don't know if there is a small correlation with the decrease in insects versus mechanical harvesting. I think that answered.
Okay, great, thank you. Another question here. In addition to collecting data on macrophyte density and species, are you able to analyze the factors that affect growth? They're giving some examples, maybe temperature, phosphate, soil characteristics, species competition.
I believe that's why the Jefferson Project came about, because no, we don't. We study specifically just biomass and locations of plants. We don't study any other parameters. I don't think Oneonta is planning on doing anything further. No. Yep, so I think that's why the Jefferson Project has come about so much, to help study these gaps in our knowledge.
Okay.
Thank you.
Great, thanks. Thanks, everybody. I want to mention that North Carolina State University has also done a plant study, a macrophyte study of some kind on Chautauqua Lake. We invited them today, but they weren't able to attend. The great news is, anybody who's talked to the Chautauqua Lake and Watershed Management Alliance knows this, but they host a lot of these surveys on their website. So if you want to check that survey out, as well as the Oneonta one, they're posted on the Alliance's website. That's it for our formal presentations. So thank you very much for all of our presenters for being here and sharing your knowledge, the work that you do. Yeah. The work that you do is obviously an invaluable resource to the community, and we appreciate you coming here to share it with us. Thanks also to Chautauqua's production crew. I'd be in the dark pretty much literally if they weren't here, so I really appreciate the help. And it's also great to be here in the golf club, so thanks for the golf club for sharing the space. And thanks to all of you. Most importantly, thanks for showing up. Thanks for your interest in the lake. Thanks for caring for the lake. Oh, sorry, I was remiss. Thanks for all the lake organizations coming and sharing what you do and all the great work that you do throughout the year. Yep, let's do it. I believe some of them may be able to stick around a little bit, so after I shut my mouth, maybe you could still talk to them or do some more networking. If you weren't able to speak to all of them, I would encourage you to do that. And yeah, thanks for being here. Enjoy the rest of your day.